Sure. So it was a session on vector development and basically asking the question how to develop safe and effective vector. And to be honest, when I was asked to chair the session, I went to the 3 talk and I was, whoa, they are all very different, all very diverse. We had someone from Oxford Medica talking about a COVID vaccine using an adenovirus. And then we had Bristol Myers person discussing an AAV platform, production platforms, and then Dino Therapeutic sharing some data on their libraries of capsid and in really deep down into how do we develop better vector. And I'm trying to find a common thread to all these 3 at first was not obvious, but while listening to the talk, really, I think we all found some really common concern related to safety and common understanding on how we are going to tackle the safety issue. I think one of the message that I heard from the first talk was that they had a platform for lentiviruses that in the midst of the COVID pandemic, they had to extremely quickly adapt to produce an adenovirus vaccines. And it was done. It required a lot of collaboration, a lot of help financially and intellectually and so forth. But it happened. So they were able to really build from a platform that a manufacturing platform that they knew well to create a new product. So that was extremely encouraging, especially in the light of what I have said, that each AAV product is a little bit different from one another. From the other talk, what I think was illustrated very well, and it's back to what I was just saying, is there's still a lot of work to be done in vector development. But this work is really feeding into the safety of the product. I think not only improving the platform, the manufacturing platform, like we heard from the second speaker, HEK and scalability and suspension to generate more and higher titer and more concentrated drug, from screening different capsids from this AAV to find one that is either more potent or can escape the immune system or that can package more fools. All of this combined creates a drug product that is definitely safer because you can hopefully inject, again, less into the human and bring less residual. So really looking at these 3 different talks, at the end really I think provided a great illustration on how all these different parts and all the different work that we are all doing in the community fed into each other and how we can learn from one platform to another one and how vector development is still really center stage of what is going to make this field successful.